Bonjour à tous. C'est un grand plaisir d'être ici avec vous for le congrès de autism. That's the end of my French for until we get to the end, but it's a pleasure to be here. I'd like to talk with you today about some new models based on our new way of understanding autism, and then some treatment trials that we're using now to try and think of other ways of approaching autism than we have been, or at least to make valid some of the more controversial ways that some people are treating autism, particularly in the United States. I'm going to go quickly through a few slides because the essence of them have been covered already in the talks this morning, and I think this fits at a good time for the Congress today to be able to perhaps pull together some of the things that we've already talked about. So I want to talk a little about novel models for understanding the etiology and the implications of this for assessing and treating people with autism. To talk more about biomedical rather than pharmacologic, I only have 20 minutes. And then discuss what this paradigm shift might mean for our assessment and our treatment of autism and where we're going in the field. So we heard this morning from Professor Bergeron that there are multiple genes involved with autism, and that while we know autism is a genetic disorder or has genetic components, we know that autism doesn't explain all that genetics doesn't explain all of autism. We've been in part impressed by what appears to be an increasing prevalence of autism. So there's debate about whether it's an actual increase in incidence or prevalence, but there does seem to be more of it. And as we understand that there's more of it, we can't explain that through it being a genetic disorder. We say there must be things in the environment that are influencing genetic expression. Some of the theories that we talk about what those could be are not definitive. There's no smoking gun for what the environmental factor in autism might be. But people say, well, maybe it's the changing diagnostic practices or better reporting or increasing recognition, increasing acceptance. But more and more people are saying maybe there are things in our environment, and those things in our environment influence gene expression or epigenetics, the way those genes get expressed. And that says something that in one way to us is concerning, saying what could it be? But in another way it says maybe there are things that we can do to influence that. We probably can't clean up the environment or take away all adversity, but maybe we can help the body become more resilient, to help it push back against what those factors are and help the body become healthier. A kind of simple way of looking at this is to say we know there's a first hit in autism that's a neurodevelopmental vulnerability, a genetic vulnerability. There's a second hit of some kind of environmental stressor that could help that vulnerable pathway express itself even more. And then there's a third hit when we say that autism is a hopeless disorder or there's nothing that we can do to treat it or to make it better. And for such a long time we did that, and sure enough autism seemed to be a hopeless disorder because we treated it like that and didn't let young people learn from typically developing peers, or we didn't try and be as aggressive as we could in thinking about things that could make things better. So we've made great progress in the third hit. We've really turned that trajectory around, as we heard from some of the speakers this morning. But we're just beginning to think about what could we do with the second, and especially with the first hit in terms of treatment. But as we do that, we've made a paradigm shift. We've changed the way. So people have alluded to the terrible part that in some ways child psychiatry's contribution to saying that perhaps there was something in the mother-infant relationship that led to autism. We've left that paradigm behind, at least in many parts of the world. People tell me that there are still some people that think that way in France, and that's sad. But people could also say that maybe a paradigm that would look at behavior, or say that it's strictly behavior or strictly genetic. We've now moved into a new paradigm in thinking about what might be involved in epigenetic expression. So you say, well, what is that? What could that possibly be? We're talking about what happens between genes and symptoms. How do you get from that? And some of the speakers this morning alluded to that as they drew diagrams showing the way the neuron might grow. But we know that things start from a basic level, but then are influenced as they grow and develop by things like immune abnormalities or inflammatory processes, which we found in autism, or oxidative stress, or disturbances in methylation, or mitochondrial dysfunction, not mitochondrial disorders, which are very rare, but mitochondrial dysfunction, those little energy cells in the body that helps the body go like kind of an energy bunny moving ahead, that those mitochondria can seem to not be as healthy, and some studies would suggest that occurs in up to 30% of young people with autism. Free fatty acid metabolism, or this morning some of the speakers alluded to the balance between excitatory and inhibitory neurotransmitters and neurotransmitter function. And there was also some allusion to hormonal effects that play a role as well. The thing about each of those is that we don't know exactly when they're happening and in which person they're happening in. So it's made us try to think more and more, how could we find a biomarker of that? How could we find a measure, a brain test, a genetic test, a blood test, that could help us know right now this person is experiencing that immune or inflammatory process, that I could influence it? Because too often we try a treatment that we think might be effective for that, and we put all children with autism into that study. And as we've heard already, there's such a wide heterogeneity that some of the people may get better, some don't, and then we get a negative study. We get a study that says this doesn't work, but we haven't pre-selected well based on a biomarker about who that'll work for. I'm going to give you several examples of that as we go through the rest of the talk today. Another way of seeing what I'm talking about is to say, what if you were to think about the Earth as a kind of model for how our brain grows and develops, or our whole body does. This summer I was with family and friends visiting Alsace, and we were drinking through some of the vineyards, and the people were telling us about the wine and how important the terroir was. And while they showed us the terroir and said, you know, it's this volcanic soil or it's this sandy soil, they also said it's more than that. It's the people that tend the land. It's the people whose heart and souls have been here doing this. It's all those things that go in to make up the terroir. When we think about the terroir that's growing a young person's brain, it's complex and it involves these processes that range from the core of the earth, if you will, the DNA, up to the symptoms that are at the surface of the earth. But we used to say the explanation was so simple that that's all we talked about, rather than saying, how do we get from here to here? And if we look at the center of the earth, that's really where the action is. That's where we can do something to change the process, to help grow healthier grapes if we're growing grapes, but to help grow healthier children if we're saying we want to affect these processes and how they're growing. And the diagram here talks about endophenotype, saying that's where we can influence things in both directions, not like we go to only one layer of the earth. If we do a good behavioral symptom intervention and we have a child that's young enough, we can resculpt neurons. We can change the way those neurons are growing by helping them learn to imitate better. If we do something that helps with oxidative stress, we can help those neurons grow in a healthier way. So another way of looking at the diagram is to look at symptoms and behavioral therapies, but looking down deeper into the terroir, one sees things like the immune function or anti-inflammatory kinds of interventions, at least theoretically. And then we can say, how could we use this then to design studies that could help us know who it's going to work for? And how could we find biomarkers to help us measure that success? And I'll tell you now about several examples as we go to the last part of the next half of the talk. One of the ways we could do this would be with pharmacogenomics, saying, could we test people's blood and look at their gene expression, that messenger RNA that was the second level up, and say, could we do that and predict who's going to respond to something that we can see so big as a medication effect. Not that I'm saying that's the way we necessarily want to go, but we can see big changes, so we could do studies that would help us know, does that work? So we had a grant from NIMH, the National Institute of Health, to study children with autism who we treated with risperidone, and we found that we treated them for 8 weeks. We studied 42 subjects, and we found that the exons of 5 genes correlated with those children who showed response in irritability and aggression. And those were genes that were there before treatment. So before treatment, we could identify 5 genes, one of which was this RNF40, which is also associated with response in schizophrenia or other disorders, to say, maybe that could be a biomarker that would help us know, would this child do well with that medication? So we put in grants to do other kinds of interventions as well. One looking at another kind of medication like risperidone, aripiprazole, to say, is it the same or different? And we're also looking at another study that I'll tell you a little more about, methyl B12, saying, could we see changes in gene expression before that predicts response, and could we see change afterwards that's correlated with those people that get better? So part of our quest is to say, is there a biomarker that we can find to help us predict response? Well, we've been doing a number of studies. Methyl B12, looking at memetine, which is an Alzheimer's drug that works on excitatory neurons that we talked about earlier. We've finished a study and have just started a second on omega-3 fatty acids. We just are partway through a study. We finished the double-blind phase of pancreatic digestive enzymes. We did a study of HBOT, and I'm going to tell you a little more about all of these in the next, I think I have 5 minutes left. And some on vitamin D, talking about high doses of vitamin D perhaps being effective. So thinking about methyl B12 and oxidative stress, how could that work? Well, there was a good study done by Jill James that said this methionine pathway from glutathione down to methionine synthase, which is the way your body gets rid of toxic load, seems impaired in people who have autism. And on the side, which you probably can't read, are a number of different parts of that pathway that found abnormalities in children with autism. There was a pilot study saying that treating those children with methyl B12 normalized their measures of oxidative stress. So we did a study when I was still the executive director at the Mind Institute, and somebody asked me earlier, are the University of California, Davis, where the Mind Institute is, and the University of California, San Francisco, where I currently am, the same place? And while we're connected, they're not the same, but I was at the Mind Institute as director for about 8 years and moved recently. So we studied 30 children there, and we found no difference in the overall group, except we found 9 children that did remarkably well. And when we looked at those biomarkers of oxidized versus reduced glutathione, we found what looked to be a predictor of who was going to respond. And based on that, we got a good-sized grant from Autism Speaks to study 50 children. And we finished about 38 now, and I hope the next time I see you, I can tell you some about our results, and that this methyl B12 makes a difference. I would tell you of the 9 children that did well, some did really well. And it makes you kind of think for a while, how could that be? Was it spontaneous, or did we really do something? One child who was 4 at the time he was in the study had been in an active ABA program for well over a year. Nothing changed, but with this injectable methyl B12, he began to acquire language, which he hadn't had before, and by the end of 6 months was told he could leave the preschool because he no longer seemed autistic. Now, I don't know whether that was spontaneous remission, whether he just got better because the treatment kicked in, but I can tell you that every child in that preschool had their parents bring their child in for the study, saying, I want whatever he got. So I don't think that means everybody should go get methyl B12. but I think it means this looks like hopeful kind of treatment that we've not been using regularly before, and with some more study, may be used for certain selected kids that might do well. We studied omega-3 fatty acids. As you know, everybody takes omega-3s. If I ask you to raise your hand, I'll bet half or more of the audience would say, yep, I take omega-3s for my heart, I take omega-3s for my triglycerides, I take omega-3s for any number of other things. There's been one double-blind placebo-controlled trial with autism by Amminger suggesting a benefit. We did a study looking at omega-3s and found a benefit with hyperactivity, although not to the point that the active group separated in significance from the placebo group. But we just got funded to do a large internet-based trial through Autism Speaks, where we could see the large number of kids that we'd need to show a small effect size by having a large number of patients. And we'll be doing that trial actually through the internet, but by mailing them placebo or active omega-3 fatty acids. Hyperbaric oxygen treatment. Has everybody heard of hyperbaric oxygen treatment? So that's a controversial one. It's quite expensive, runs into a lot of money. You need to have a chamber, and people say, does it work? There are 2 double-blind placebo-controlled trials saying it doesn't. One placebo-controlled kind of trial saying that perhaps it does. We said if it works, it might work by changing inflammatory processes. Let's look at cytokines. So we measured cytokines in 10 children, and amazingly, none of them had abnormal cytokines. We thought half would, half wouldn't. We did the study, 9 out of 10 parents said their child was a lot better, but it was open label, and that's not a good trial design. But we didn't find that cytokines were routinely abnormal, which are a measure of inflammation, nor did we find that they changed as a result of HBOT. Namenda, memantine, this medication that's used to treat Alzheimer's, works on excitatory neurotransmitters. It's made by a company called Forest, and they're doing a double-blind trial at 8 or 12 different sites in the US, trying to see if it helps with autism. There were some open-label trials that suggested it's a benefit. It's a relatively expensive medication, and I think they say that things are looking good for at least a small group of children for getting better with Namenda. Probably not for everybody, but perhaps for a subgroup. We just finished a trial with a pancreatic digestive enzyme. The idea of the casein gluten-free diet is that people's bodies who have autism have a hard time digesting large proteins. And so they go through the gut, those large proteins, undigested, and cause inflammation of the lining of the intestine. Some say maybe pancreatic digestive enzymes could help break those down so you wouldn't have to go through all the effort of a casein gluten-free diet. So far, we've seen some kids do very well. We haven't broken the blind, but some kids seem to be doing well in the study, suggesting maybe there are a subgroup that have GI problems. Increasingly, we're thinking of autism as not a brain disorder, not a mom disorder, not a brain disorder, but a whole body disorder, a disorder that affects your GI system, that affects your immune system, that affects your brain. And how we can use that whole body kind of focus makes a difference in how we might be able to get improvement in the symptoms. Finally, we're doing a vitamin D study that's right now at our IRB, the board that approves studies. We're in our second round with them because we're using fairly high doses. And the concern with very high doses is that you can decrease calcium levels, increase calcitrol. So we're using those as biomarkers to measure that effect. But there are anecdotal reports of some young people doing very well with high doses of vitamin D, children that have autism. So the hope would be that we're pushing back at resilience, that we're helping these young people's bodies become stronger at withstanding that second hit, whatever is interacting with that vulnerability, and helping that gene expression work out to be as healthy as it might be. So when I think about practicing, and I take all those things into account, and this is my second to the last slide, I'd say that first I start out thinking about what are the medical things that might be playing a factor here? How can I think in a whole body way about this disorder of autism? So right now in the US, it's recommended that we get genomic microarrays, CGH arrays, on all people with autism. It's maybe not standard practice, but it is looking at those arrays to say, could we see, like Dr. Bergeron this morning talked about copy number variants, talked about all the other things that relate to gene expression. Could we see those abnormalities, even if they don't tell us exactly what's not working now? They give us a baseline to work from. We think of having a good neurologic part of the evaluation, especially since 30% or more of young people with autism go on to develop a seizure disorder. When they have GI symptoms, we think about getting a GI specialist involved, although many of them don't care much for autism. But we still think the GI system is not unrelated to autism, which is what many GI specialists tell parents when they come in with their child who has GI problems. And we take seriously all the medical symptoms that people have. We think about the importance of their getting speech and occupational therapy. Behavioral treatments are the ones that we have the best example of what works, and it's important that people get into good behavioral programs. And you should know that ABA is more than discrete trial training. It's a variety of applied behavioral interventions. Treat associated symptoms, and sometimes that involves using pharmacologic agents. And then think about biomedical treatments. Omega-3s, I suggest those to most parents. Melatonin, I think works great for children that have sleep problems. There's a few other things that are kind of standard kinds of things that you could do that aren't outrageous, that can make a difference in how children develop. They're often subtle changes, but they're enough over time to make a big difference. This was a slide drawn by a young person who had autism and didn't express himself very well. His mother said that he would always draw a situation of conflict and then show a way out of it as his way of expressing himself. This picture that he drew is called the haircut. You can see how he feels about getting a haircut. You can see the man up there with the scissors cutting his hair, and the bee is probably the buzz of a razor cutting his hair. So over in the corner, you see a little door that's his way out of this uncomfortable situation. I hope that by this paradigm shift, by thinking about autism as a disorder in some ways that we can help by improving resilience, that we shift the paradigm in how we begin to understand autism, and we can help young people find their way out of this uncomfortable situation. Merci à Monsieur Sajidi et Amélie et tout pour cet génial congrès. Merci pour votre accueil.